There we go. All right, so that was perfect time to restart recording because I'm going to start talking about quickly mentioning who our two presenters are tonight. First, we have Reese from Oxen Games, and we have Nigel from Myomics. So with that, I am going to spotlight Reese. And as he gets ready to present, I'm just going to give a quick overview of Reese and Oxen Game. Oxen Game is a game design studio that creates quality, attractive, and fun educational video games, and redefining the standard definition of a, quote, educational software. So with that, I'm going to hand things over to Reese.
Hello, everyone. Yes, I am the one student from MSU, and I see all of the U of Mers, so don't hate me. I'm going to quickly share my screen here. Let's hope it works. Okay, can you see everything? Good?
Yep.
Okay, great. So hello, my name is Reese Jonas. I'm going to pitch on my company, Oxen Games. Of course, there we go. Okay, so I'm a student studying microbiology here at Michigan State University. And I'm actually the first person to attend college in my family, making me a first generation honor student. And so education wasn't very well understood or attained in my family, but I found it to be so intriguing and I fell in love with it. Obviously I'm here, and particularly with biology. So I want to learn more about education and the pitfalls that exist. So I did a little bit more research and found a huge problem. The average science test scores in 12th grade, in this case, and interest in science hasn't changed. And in 2009 to 2015, it remained stagnant right above that basic level, which is a huge problem. And so to combat this, science educators across the nation developed these new novel science education standards, and they're being implemented into many states across the nation, about 2/3 of the nation. And what's important here is that these standards focus on a different way to teach science, a model-based approach instead of like a lecture-based approach. So instead of students asking questions, it's reversed, and students pose questions to the teachers. And so because of these new standards, teachers, schools, and students need new tools in order to help teach this up-to-date method. And so we created infection defense. We kind of started off with this idea and we piloted it in about 20 high schools and have implemented it into a couple outreach programs at Michigan State. And so infection defense is a video game about the immune system, taking a starkly different approach than traditional teaching methods. It's interactive, appealing, and high quality, something that education really needs right now. And I'll show you a demo later, but a lot of the teachers and students that we've tested this product with really love it and engage with it so well, and has the capacity to completely replace textbooks. And so this is where we want to go. We want to completely replace the textbook and create software similar to infection defense that has the capability to teach entire courses, not just single disconnected concepts like the immune system. The textbook, assessment, simulation, interactivity, and the online nature all merged into one platform. And so we know we can apply this gamification technique to other biological concepts to an entire course because we've done it before, such as this game, Cascade, game about phosphorylation cascades, and we've designed other games, plant communications, et cetera, et cetera. And so what we want to do is take this gamification technique and apply it to this entire course. And so where will we start? How will we implement this? How will we make money? And so what we want to do is approach these textbook companies and use our software to accompany their textbooks. These textbook publishers are attempting already to build out their electronic platforms in order to keep up, not only with COVID and the online push, but the advancement towards technology in general. And we see a potential financial model of a simple license and royalty fee for every distribution that the game and textbook that they distribute out to every student. And so what we want to do again is take all of the current alternatives to education, maybe the free videos from YouTube, the assessment features from Quizlet, reading resources from various textbooks, et cetera, and merge it into one platform. And so what I want to do here is show you a demo of how this works with our current game, Infection Defense. And so what this game does is it takes you inside of the skin and you are interacting with the different white blood cells. As you can see here, it's showing you how exactly these white blood cells, what effects they have on us and what they're actually doing. In addition, there's a full textbook inside the game that students can go and read about, for example, a blood cell. There's also an assessment quizzing feature, incentivized learning, where students can answer questions and receive kind of in-game currency. And so we really see this as a complete replacement to education. And what's interesting is this is kind of already a technique used in classrooms right now, is they kind of will just show pictures of the skin just like this, but we are taking this and we're making it dynamic.
Oh,
I think it might be happening.
anybody else?
Can you hear me?
Reese?
We can hear you again now. Yeah, there was a minute where you cut off there.
Okay. Can you hear me now, or
Yes.
are you able to see my screen or no?
No, I think you have to reshare.
Okay. Okay. And yeah, let me reshare it. Sorry, my internet's not the best right now. Can you see my screen? No.
Yes.
Okay. Well, that was my last slide, so it doesn't so matter. But I really want some help, whether it be another team member or potential advisor, in my go-to-market strategy. I really want to get this out in front of the right people and establish the right connections, whether it be in STEM education realm, education technology experts, or other individuals that are involved in publishing company realms. So that's really, if there's anyone in the call now who is involved with this or has the right connections, I would really like to speak to you and get some help. But thank you all so much for watching. I'm sorry about the technical difficulty there. And if you have any questions, I would love to answer them, or any comments about anything at all. I know
Yeah.
Oops.
Well, as folks are thinking about their questions, feel free to type them in. I'll just ask the first one that I have. Where did the name come from?
So auxin is actually a plant communication molecule, and in plants it stimulates growth, which is kind of the image I want for the company, is to kind of emphasize this growth factor of learning. Oh yes, and there you go, the links in there. And so Diane, yes. The question is, have you tried to gather any data on information retention? And yes, I actually currently am. So we're doing research here at Michigan State, me and a couple other professors on the game itself. And we're just basically distributing a control group, just a traditional article that students would read. And then we're giving, a different group the game. And we're kind of just analyzing pre-post, what the results are there. So yes, I am. Thank you for that. And Roger, yes, I'm studying microbiology and molecular genetics. So it's the immune system, what you saw there. And I particularly love pathogenesis, which also ties into this game. And I was learning this in my classes, and I really found it interesting to me. And I saw a potential to gamify it and visualize it instead of just this traditional lecture format. And so Robert, can teachers evaluate students? That is one thing that a ton of teachers have been asking about. And one thing that I'm putting in, the update will come out within the month, is a dashboard. So those questions that I kind of showed you in that demo, it'll be able to get all that data, all the questions that they answered correctly, the ones that they answered incorrectly, and export it to this easy to read dashboard. so teachers can see which students are answering correctly, what they're understanding, what they're not. And then grades are, so I may have missed the grades you're targeting. Have you done any primary research with teachers and classes? Yes. So it's in about 20 different high schools, as well as a couple outreach programs at Michigan State. And it's going to be distributed to high schools across even more high schools across the state. And yes, I've actually, a lot of teachers have let me come into their Zoom room classrooms and talk to every single student and the teacher, of course, into what exactly they like and what works for the teacher, how this is actually educating our students more interested in STEM, in the immune system, things like that. And any inquiries on application of COVID? I'm glad, Jim, you brought that up. So yes, and what I really want to do with this as well is apply it to vaccines. There's a lot of kind of this stigma on vaccines, and either they're unsafe or not effective, or they're putting microchips in our bodies. And what I want to do is actually incorporate that in the game. And it can easily be done where you can get a vaccine and it helps you in the game, and kind of visualize and show what vaccines do. So yes, that's something I really, really want to do, is apply it to current problems, whether it be COVID, the actual virus itself, or vaccines. And Stefan, do I have a co-founder on the technical side? In the beginning I did, they just got caught up with school and stuff. So currently it's only me working on a lot of stuff, which is why I really am looking for someone who wants to do something like this. Anyone, particularly, someone with marketing skills, but also technical skills would really, really help. And then another question, but rather a referral to a former neighbor who is a textbook publisher. Yes, Maria, I would love to speak to them, and I can put we're going to be in the breakout rooms, is that correct?
Yeah.
After this?
Yeah, it's going to be randomized.
Okay.
So you just want to share your information below, and that'd be really great.
Okay.
And we have about 40 seconds left for Reese or any questions. And even if you think of one later, feel free to chat it in the chat, and Reese will respond to chat when we move on to
yeah. Thank you. Yeah, I put my email in there, my school email, and I also put the link to the website. So if anyone wants to actually go and play the game, it's not as updated as the one I showed you, but it's still very workable. Actually, yeah, that's great. I'll copy that email. Maria.
Okay. It sounds that's great. You had an ask and you got got an answer. That's always kind of great there. That's what we hope for here. And Reese, thank you for, first of all, taking the time to present tonight. Really great work. Super, really cool. And I'll probably try playing that game myself a little bit and refresh my microbiology knowledge. With that, I am going to spotlight Nigel so he can share with everybody Myomics. Myomics develops web-based data analysis solutions for single-cell sequencing and other high-throughput omic applications. Our products enable experts to perform their own data analysis without the need to master complex bioinformatics tools. So with that, I'm going to hand things over to Nigel.
Great, thanks a lot, Hein, appreciate it. Sorry, the screen may be a little bit small. I'm visiting family and I'm on my tablet. But I'm really excited to talk to you today about my CV, which is not my resume, but it's rather the first product that is coming out of this company we're calling Myomics. We're a SaaS company trying to solve really complex genomics problems for biologists who are working at the bench doing their own experiments. So over the last 5 years or so, this class of experiments we could call single cell sequencing based experiments or single cell technologies have really changed the game for biologists because they've enabled biologists to study what's going on inside of cells at a previously unprecedented resolution. Essentially, instead of looking at big test tubes full of cells and kind of mixing together all the data that came out of them, they can instead look at every single individual cell within that tube and look at what's different or the same across all of those cells. But while biologists have recently been able to take advantage of the commercial and technical support that's necessary to physically carry out these kinds of experiments, the data that's generated on experiments like that can look something like this matrix over here on the left. It can be gigabytes in size. It is representative of the expression of a wide range of different genes across hundreds of thousands of cells. And on its own, this matrix on the left is really not useful. On its own, you can't really make anything from this data and form new hypotheses from it. What biologists need in order to actually act on the data that their experiments generate are plots that look something like this one on the right. It's one of many different kinds of, we would call like final plots or final statistics that can be generated from that matrix on the left. And in order to actually generate plots on the right from data that on the left, it can easily take a novice who's just doing this for the first time hundreds of hours to learn how to do it on their own, because the tools required to do it are still stuck in the command line. People have to go back to their command line terminal and type in commands that can take a long time to master. So what some biologists do, they could hire themselves for free, but it's going to cost them hundreds of hours. Many biologists will actually hire an analyst. They'll outsource this work to an analyst. But through a lot of customer discovery, both with biologists, with analysts, in my own work as a consultant and a grad student, what I find, what we've found is that both analysts and biologists find this kind of data sharing to be pretty frustrating on both ends, because single cell analysis in particular is a very iterative process. You don't just make this plot on the right once. You're going to make it once, you're going to look at it, you're going to ask the analyst for more information, and you're going to go back and forth generating hundreds of emails. There's a lot of solutions to this problem. None are particularly streamlined. And so our solution then has been to develop a web tool that enables researchers to upload this data matrix on the left that they generated themselves, click a single button, and 5 minutes later, have data that looks like this thing on the right that they can actually act on. Let me move on a little bit here. So this one-click web tool, there's a little screenshot of it on the right. This is a pretty minimal deck that I've been using lately, so let me know how it goes afterwards. But it looks a little bit like this panel on the right, and it essentially, after clicking that one button, lets researchers download a printable summary report with all of the plots and statistics they would need to even publish this data set. It comes with interactive tools where researchers can go in and interactively select cells of interest, explore external databases to learn more about their data. All sorts of things that normally would require a lot of hands-on effort can now be done in single clicks on a web tool. And throughout the web tool, we've tried to explain all of these steps, all of these plots and statistics in a plain language, because again, through our customer discovery, what we found is that both analysts and biologists are finding that this data analysis space is so complex that it's tough to even find a common ground to talk about it on. And so by trying to explain this to biologists who aren't data analyst experts, what's going on at a level that they can approach, it makes everyone more efficient, regardless of whether or not they're even going to use this tool for their final publication. Products like this, there's no question, they sit in a pretty small technical niche. There's probably very few single cell analysts out there in the audience today, and even across the globe, there's only 1,000 to 2,000 perhaps. But nonetheless, we've tried to see if there's a market case for this, and we think there is. If we look at how many single cell experiments were done just last year, which is more than 100,000, and we consider the amount of time it would take a very competent and trained analyst to do a basic analysis like our tool can do on their own, we find that nearly $160 million was just spent in 2020 on this kind of analysis. on this kind of analysis. And in a market that's growing 15% year over year, what that means is that by 2025, it'll be 320. So for a SaaS product that's cheap to develop and delivers real value in this space, there's definitely some opportunity. Our plan to push this into the marketplace has been focused on getting this tool into the hands of individual researchers in academic research labs right now, as opposed to directly going to corporate partners or academic sequencing cores, which are our eventual targets. A lot of our customer discovery at that level has shown that getting the independent researchers to buy into tools like this from the top down, from a sequencing core down to their clients, is really challenging. Whereas if we can get a lot of the individual users to start using the tool on their own and lobby their institutions for larger licenses, that tends to be a more effective strategy. So we're trying to get into the labs with these hands. We've got our beta out to more than 5 labs here at UM, a full sequencing core at Ohio State. We now have a lab at UC San Francisco that we're working with as well. And while we're not growing super fast just yet, I'm doing my best to put pedal to the metal to get this into more and more people's hands. And the big ask here, I guess, is to see if there are people in the audiences network that may either work with people who do sequencing or who work with companies that are in this space that may be interested in expanding their portfolio of software tools to include something like this, because that's where we're starting to try and spend a lot of our kind of interview hours or interview efforts, is kind of expanding our network in that space as we look to the future. I guess that's kind of the end of it here. So my colleague Logan and I'd mentioned briefly at the beginning, we're both single cell biologists. We've been doing this ever since single cell technologies came out about 5 or 6 years ago. So we've been around in the space for as long as it's been around. And we've been working here at UM alongside Ann Arbor Spark and with the Venture Center to try and, I guess, as biologists who work full time at the bench, learn about how to bring a SaaS product to the market. So we'd love to discuss with anyone in the audience about their experiences in the space, get some advice. And with that, I guess I'll end and ask for any questions or comments. Thank you, guys.
All right. Thanks, Nigel. Appreciate the pitch and sharing. Looks like you already have some questions, at least a couple questions from Jim here.
Since I'm on my tablet, I'm going to stop sharing so I can go to the chat window, but I can go back if people need that. So Jim, you have a question about applications to COVID effort, excuse me. There definitely are people who are studying COVID-19 at the single cell level. Their data sets are just as applicable, or I guess our tool can analyze their data sets in the same way that other pipelines that we are trying to essentially replace or augment can. So there's no question that we can work with those kinds of data sets. And one of the things that our tool, we've spent a lot of time adding in, is linkouts to external databases where, in essentially real academic time, as research enters these national databases, we, on our tools end, can pull up data from those papers, from those databases to supplement the data that the users are providing themselves. So if they, in one of their COVID samples or any other sample, they see that gene A is really prominent in one cell type. When they click to see what gene A's expression is, they're also going to get a database table that shows them what gene A's name is, what its function is, what new papers have been published about it, what people are talking about it, so that they can add that context. And you also have a question about, do you translate statistical concepts into research biological terms? I think that the answer to that question is yes. So I've essentially spent a lot of my own time trying to break down this really complex statistical analysis pipeline into intuitive diagrams and plain English language descriptions. Because the math that goes into a lot of this analysis, while complicated at the mathematical level, intuitively it's not that complicated as long as you've got a drawing and a somewhat intelligent description in front of you. So biologists really can wrap their hands around it if they have that information available, and we're trying to provide that. Neil mentions TriSphere Ventures in Champaign. They made money there with flow cytometry and Sony. So I will definitely look into that. Thank you, Neil, for the mention. Robert's asking, what's your revenue model? So we are currently selling this as an individual user subscription service. So users can go into the tool, they can play with the demo for free with data we put up there, and then they can currently purchase an independent license, one user at a time. Our plan is to get enough independent users on at a pretty cheap subscription rate to then eventually ramp up the price and push for site-wide licenses where UM's advanced genomics score will pay for an annual license to our software on our infrastructure for all users at UM. That's a model we've seen work with some other bio SaaS companies. BioRender is starting to go that way, which is pretty recent and seems very successful. So we've seen precedent for that model, and that's kind of our strategy. Hope that answers your question. Andy is asking a question. So bioinformatics is known for very messy file formats. How much data transformation is needed before people can use their tool? That's an excellent question, Andy, and you're totally correct. So there are very messy file formats in the general bioinformatics space. Luckily, in the single cell space, at least as of 2020 and 2021, there's really one company that is dominating when it comes to physically performing these experiments and generating the raw data that our tool takes up. That company is 10x Genomics. And what we essentially are leveraging is the fact that they have a very small subset of file formats that they use that we can support very rapidly. So that's part one of my answer to your question. Is there's a pretty limited subset in this specific case. The second subset or answer I want to provide is that we're actually building on top of a wide range of open source pipelines that already have importing tools available for many of these messy formats. And so we're not really reinventing the wheel here for data import, for data analysis or anything. We're really providing the graphical wrapper and the extra fixings. So even as these formats change and some of the needs of our users change, we might have to do a little bit of manual coding to make some of those changes available to users, but by and large, by building on top of open source software that already handles these issues, we stay pretty lean on our end as those formats change. Hopefully that answers your question. Maria is asking if I work with anyone at UM Hospital and surgical innovation teams. No, I do not right now. So if you have, I guess I'll do some searching there to learn more about what they're up to. But if you have any specific contacts you think I should be talking to, I would love to hear more about that. Yeah, I'm still trying to expand my network here at UM as well as around the US right now. So please, would love to connect. Thank you. Steve mentioned, so since I say there's only a couple thousand single cell scientists, it should be pretty easy to contact them and show them your product. The answer to that, or I guess my response to that is that it should be pretty easy, yeah. I am building a list of them. I am trying to go around and build up that contact list and doing some cold contacts. It's just, we've only recently gotten our product out there in the format we're happy to show to people. So as I finish up my PhD thesis, I'll continue to reach out that way. But you make a great point. It is a pretty small group of people, so it should be pretty easy to track down. I think that's it for now. Any other questions or thoughts? Those are all really helpful and really informative. Thank you.
Yeah. Great. Cool. And yeah, if there's any other questions for Nigel, feel free to send in the chat, or if Nigel, if you want to put in your contact information for folks too, if they want to reach out to you.
Absolutely.
But thank you for taking the time to share Myomics with us. It was really cool to find out what you're doing.
Thank you for the platform. Appreciate it.
Yeah, and as I mentioned earlier, unfortunately, our third pitch couldn't be here tonight, so we can all wish him well as he recovers from COVID-19. But hopefully, in the next couple of months or so, he'll be able to pitch. And we do have a good, just to kind of quickly pitch for our schedule for next month, which is also going to be on the 16th of March, 3 months in a row where we have our meetup on the 16th, which is kind of funny. We have a few good pitches there already, so definitely come and check it out. But if you are interested in pitching in the future or know anybody who should be pitching, feel free to send them my way, or you can contact me at my email, which I will send to everybody here. And yeah, so just some general criteria as you've kind of seen here is that like the product should be beyond kind of like ideation. There's some at least some initial kind of testing to validate the concept. And with that, I'm going to open things up for folks to just chat and type into the chat any kind of community announcements that they might have. So if you are looking to promote a meetup that you have, a position that you're hiring for, anything like that, feel free to type it into the chat. And what I'll do is I'll copy and paste it into the meetup page just so it lives in prosperity. But we'll open that up for a few minutes, and then from there we'll do some breakout sessions. So we have a few announcements in here. MGCS, extend your application deadline. If you're fundraising, apply. 2 is hosting a clubhouse room on EV this Thursday at 8 a.m. Eastern. They'll be talking China, EV, AVs, and mobility. That's pretty cool. And Diane may know of a couple of U of M affiliate startups hiring developers. And 2 provided the information where you can find them and the clubhouse. David mentioning he's launching a new analytics platform for attorneys and are looking to hire a commission-based salesperson. Okay. Any other last-minute community announcements? All right. Well, with that, I want to thank everybody for, like always, taking the time to come out and supporting our pitches and supporting the meetup. Again, the next one will be on March 16th. What I'm going to do now is just break everybody out to breakout sessions.